We're going to launch the Roundtable. So we're going to continue talking and thinking about all the incredible things and problems, issues that were told earlier in the morning with a great panel and with also the participation of industry, which is always very important and interesting to see the bridges between the two and, of course, the strategies. But to connect to what we've just said before, I'd like to start again with you, Emmanuel, and it will launch the rest of the discussion and, of course, questions and answers after our first exchange. You talked about many important issues and things, but maybe you could focus on toxicity. You just said a few words about it, and I think that it's one of the main issues that you face. Neurotoxicity, cytokinic storms, and others. Can you tell us more?
Well, thank you for the question. Is it working, the microphone?
Normally, yes. Just
yes.
try it.
Oh, thank you. No.
Okay.
Oh, no,
thank
it's
you.
fine. Thank you. Yeah, no, of course, this is a very important issue, the toxicities. We know that there are, as you said, different kinds of toxicities, those mediated by an exaggerated immune response, a cytokine release syndrome. But this one, I think the clinicians have learned how to manage the cytokine release syndrome, IL-6 production, and neutralizing IL-6 and its effect. There are also the neurotoxicity, which is also, it was not predicted at all using the current preclinical model, the mouse model that scientists were using. We missed totally the neurotoxicities.
How can you explain it? We have an idea?
Because simply the current model that we are using testing the efficacy and potential toxicity are not the good one. For the moment, yes, that's right. For the moment, we are using immune-deficient mice transplanted with human tumors and infused with human CAR-T. And in these animals, we missed totally both the CRS but also the neurotoxicity. And so clinicians were quite surprised initially to see such severe toxicities in some patients. And I have to say that I think, I'm not a clinician, but in terms of mechanism underlying responsible for this neurotoxicity, still a lot to learn. Is it due, for example, to the infiltration of CAR-T into the brain, to also the diffusion of inflammatory cytokines to the brain, or eventually there was that paper from UPenn showing that CD19 could be also expressed by a very small fraction of brain cells, the neural cells, which are covering the vessel. So eventually.
So it could be targeted.
That's right. So again, still a lot to learn. And yeah, I mean, of course, we need more predictive models.
So you work on humanized mice? That's right.
Humanized mice could be one model. We are working at Cochin in CERN on another model. Actually, I presented the model with tumors this morning, slices made from human tumors. But we are also developing the same model also for a European IMI consortium, T2Evolve, with healthy tissues, slices from healthy tissues. And so we apply CAR-T cells onto healthy tissues, slices from healthy tissues, and we looked at potential activation of the CAR-T and destruction of healthy cells. Because I forgot to say that another important toxicity is the so-called on-target of tumor toxicities. Mm-hmm. The fact that when the target is expressed also by healthy cells, even at very, very low level, tiny levels, of course the CAR-T cells, they don't make the distinction between the tumors and the healthy cells. It is what's happened actually with the commercial CD19 CAR-T. We have a total destruction of the healthy B cells. But when the target is expressed by vital organs like the lung or the heart, then it could have very, very severe side effects. So we definitely need to have a better model in order to predict such toxicities.
So great challenge among others. And of course, one of the most important is to have people more responding. You have 20 to 30% of patients who are responding, but all the all the others don't respond. So that's a very important issue. And Sante Kundari, you are Senior Director for Gilead, Head of External Scientific Collaboration in Medical Affairs, Oncology and Cell Therapy. So, of course, you address this problem. You were one of the first companies to work on CAR T cells. I think it was 2007. It was early, very early. And now you want to address this problem, especially, which is one of the most important issues, on solid tumors. You talked about it a lot this morning. Solid tumors is very important to reach today. And you work on application multiple myeloma and others. But solid tumors is one of your most important targets.
Yeah, I would say that we are continuing to work always in hematological malignancies. That will continue to be our, let's say, main goal for the near future. But we have some assets starting in solid tumors, especially in brain tumors, thanks to a collaboration we have with Immunity and Penn University. So we would like to address and to target glioblastoma and neuroblastoma, also with a focus on children. It's really early stage in terms of assets, but is one of our, let's say, future things to look and to see in the future. With all the, I would say, challenges we discussed before, because solid tumors is really different for the liquid, I would say, cancers. But again, we hope that first, having the right target, that could be one of the issues we already discussed, could be one of the things that I think from a sponsor and company perspective, we want to see if the right one. And then, yes, finger crossed that we are working hardly on that. Let's see how we will work.
Okay, so we'll go back to the strategy and maybe autologous and allogeneic approaches, which are also another issue that you all share. But on the same topic, Vigo Ventandolu, oh, sorry. I was just saying, but the photo is not the good one. Oh, okay, sorry, I've got to make a round. Sorry. So, Vigo, Director External Innovation and Benelux for Johnson & Johnson. And you also addressed the question of solid tumor, which is a very important issue for you too.
Absolutely.
No, it should be working now.
Yeah. One. Yeah.
Okay.
Yeah. Sure. No. And just as Sante said, the main focus, of course, of Johnson & Johnson is still to develop CAR Ts for the hematological malignancies. But of course, we're also looking very intensively to the field of solid tumors, as we are aware of the bottlenecks and obstacles that are still out there. And I think they can only be tackled by what I would call synthetic engineering of the CARs in order to arm them with potencies and features that could withstand this tumor microenvironment and able to invade the tumor and exert their function. So I think we're not there yet for sure for solid tumors. There's a big road ahead for us, and I don't think we can solve all the problems, but I think if we can combine efforts and we can collaborate, I think that would be the goal for solid tumor CAR T cells, and use the knowledge that come from academia and see how we can apply that knowledge in a translational way to make ultimately to come up with better treatment for patients. So, yeah. All
right, and so you have different strategies, and you think about some specific issues. For example, current therapies are focused on alpha beta T cells, but you say there could be an interest to focus on gamma delta T cells.
Right. Next to engineering, let's say the classical CAR T cells, I think we should be open to other possibilities of not only T cells, but also looking at other cells. But even within the T cell population, I think there's a lot to be learned on which T cell subset we should aim to arm with a CAR T, and not just take the whole population of the T cells. We have indeed a population called gamma delta T cells, which are known for their intrinsic cytotoxic effect. So if we could arm these T cells with CAR Ts, with a CAR T construct, I mean that would make sense, of course, to make even a better or a more potent CAR T.
Another issue that we'll, of course, share together is that you'd like to move from oncology, which is our main focus, to autoimmune disease.
I think many people are now looking into the possibility of moving from oncology to other domains as severe autoimmune diseases, as we heard this morning from Novartis, to use CAR T cells as a way to deplete the autoreactive B cells. So indeed, I mean, it's a very young field in its infancy, of course, and we need, of course, to really outweigh the risks of CAR T and the manufacturing challenges, of course, for CAR T in that area. But I think good strides are being made to also use these CAR T cells in autoimmune disease.
All right. So now, Alessandro, if I'm not mistaken, Alessandro Crotta. So you're global program lead cellular therapy development at BMS. You've already got 2 products approved in that field, Abecma CAR T and anti-BCMA, that's right. And you work on multiple myeloma too. And you also want to, well, you focus on cancer, but you also, like, as we said, focus on immune disease.
Yeah, correct. So at BMS, of course, we have deep knowledge on what CAR T can do in hematological disease like lymphomas and multiple myeloma. But now we are focusing also, as many others, on autoimmune disease. So we have ongoing trials in different autoimmune disease. That's the beginning, because of course we heard this morning about the data in lupus. We are looking at other disease in autoimmune that have autoimmune background, like also neurological disease. So it's a quite large area that can be investigated where there's a potential for CAR T cells.
All right. The question of the production of these CAR T cells and the use of patient cells, you say that maybe there could be an interesting issue with iPS cells?
Yeah, I think we already see the limitations that we may have with autologous CAR T cells, because it's a very complex process for each patient. So we need to find ways that make more efficient and scalable the production of these cell therapies for patients' populations that are larger and larger. And of course, autoimmune disease, it's a first example of a very large patient population, much more larger than what we are now treating in hematological disease like lymphoma and multiple myeloma. And so for that reason, we look at other platforms that can be used, specifically allogeneic cells that can be donor-derived, but also iPSC that are an interesting platform to be used for expanding the production from one single source for multiple batches.
Okay, so it leads us to that very important question, autologous versus allogenic platforms. And there is another way. Karine Rossignol, you're CEO of Start Immune. You were one of the co-founders of Imagine Foundation with Professor Alain Fischer that we'll listen to this afternoon, namely. So you're a great specialist of the question for many years, and you propose something that could answer that problematic, which is pro-T cells, which would avoid all the situations we want.
Yes, indeed. So we are developing a new T-cell subtype, the T-cell progenitors. We can differentiate in only 7 days ex vivo hematopoietic stem cells into what we call T-cell progenitors, meaning T-cells which have not been educated on a thymus yet. And the interest of that is that we can have a kind of the best of the two worlds, meaning we can use HSCs coming from an healthy donor, differentiate them ex vivo, and then infuse them to the patient to be treated. And his own thymus is going to differentiate those cells into a fully polyclonal new T-cell compartment. And those cells could express CAR T. And in that case, we could have CAR T, which could have a much longer lasting effect, because indeed educated by the own patient thymus.
Okay, what's the stage now? Where are you?
So we have validating the concept and the rationale in hematology post-transplant. All the data are pointing in the right direction. We have demonstrated that we can rebuild a fully functional T-cell compartment in severe combined immunodeficient babies. We are at early stage, this is only 10 patients included, but demonstrating the same in adult leukemic patients.
Right.
So we have a project which is still in preclinic on off-the-shelf CAR Pro T-cell, which is very exciting.
Great. Well, Emmanuel, what do you think of this approach, allogenic logics, and this way?
No, I mean, as I said, I mean, conventional T cells, alpha beta T cells, they have many advantages. You could produce them in large numbers, but also they have some drawbacks, limitations, especially for an allogenic perspective. If you want to use an allogenic T cells, you will have to manipulate genetically that T cell. That could be very complicated. So instead of doing that, of course, using other cell types such as NK cells, gamma delta, NKT cells or other cells or iPSCs would present a very important advantage, of course. It makes sense, and I'm sure we'll hear a lot about developing some immune cells equipped with a CAR. Right. And also including, and I know that we have also experts in the audience, some innate immune cells equipped with a CAR. It could be monocyte, could be macrophages, it could be B cells. So we are lucky we have all experts in the room on these aspects.
Okay. Right. On this issue, on this autologous versus allogenic approach, what's your point?
Well, I think that, of course, everyone now is especially in the companies, I mean, and Johnson & Johnson in particular, we are focusing on these autologous products. But of course, we see these enormous manufacturing challenges to supply all the CAR T cells for those patients in demand. So we need to work on a solution in the background, of course, to be able to treat every patient that needs that therapy. So, and I think then allogeneic, and even if I can go one step further, even in vivo CAR T cells could be one of the solutions. But of course, I mean, much work needs to be done there, and it goes without saying that off-the-shelf CAR T strategies do make sense from a logistic point of view. To have enough batches and CAR T cells ready for the patient's demands. But again, we face some immunogenicity problems with the allogeneic CAR T cells. We can engineer them, or we can re-educate the T cells again. So I think there are some solutions on the horizon.
Okay. Do you look at it, something?
Yeah, we are working. We are working on allo as well. I think we have some assets in early stage as well, and obviously, personally, I don't see auto versus allo as a fighting. I see how the asset can be complementary and also helping some of the issues of the others. Obviously, on the paper, allo it represents a big hope, I would say, bypassing problem of having the asset and the treatment ready for every patient in needs, et cetera, et cetera. Still, I think we have some issues, and we just heard that there are some solutions of it. Cell gene editing is helping to avoid some of the big problems for GVHD, for example, that we can have for the allo as well. To me, and this is my personal thoughts, is also to verify how the scale-up of the allo will be in the future, because the industrialization of the asset then is very important. As a company, we are working, let's say, how to improve the autologous asset and just to reduce the timing, I would say, of the product to the patient. So we are working to automations and to work to produce and to restrict the time for the auto. Again, it's complementary, is absolutely a very good hope. Still some challenges, I would say, but we are close to recovering to save, I would say, these challenges.
Yep. Okay.
Maybe also what is important is the quality and the profile of the cells. So if you take autologous and you use T cell of a patient which has undergone a lot of treatment lines, those T cells are going to be damaged. So they're going to have a hard time to provide, even with a CAR, the efficacy expected. If you think about it, iPS is definitely the way forward. I think we are not there yet for T-cell application, because again, a T-cell is supposed to stay in the body during 7 to 10 years. So there is obviously tumorigenicity, safety to be challenged there.
Related to the APS.
Exactly. So at Smart Immune, we do think that going first by using T-cell progenitor is one step in the right direction. But of course, on scalability, iPS would be definitely the ground, and we are all pointing in that direction. But again, quality of the cell is key. to overcome exhaustion hurdles. Sorry.
All right.
I fully agree with what has been said, but maybe another aspect which is important when we talk about allogenicity, is the cost. Of course, it will decrease, and I think I'm sure we will discuss about the cost of the product. Yeah, but having an allogenic product will, of course, decrease the cost of the treatment.
So a treatment is around about $300,000, I think. It depends, of course, on treatments, but the prices that we can see here or there, you've got two products. And of course, there's the problem of targeting the patients. If you only get 20 to 30% of responding patients, how do you target them? Do you make kind of a companion test, or do you use imagery to try to follow the distribution of the CAR-Ts? How do you select your patients, or do you know how it works in clinic?
Yeah, so this is a $1 million question, I think, that everyone has on which are the right patients to treat with cell therapy. And of course, we are, as of now, I think with the experience that we have with the different products already on the market and where there's more experience even in the real world setting, I don't think that there's any signal for now that provide a clear answer to this question. There's still a lot of work that can be done and is ongoing to clearly identify those patients that benefit best out of the available treatments. And of course, especially when you move, you want to give this kind of therapy in earlier lines, this is more and more important because there are standard treatment that works very well for some patients, but not for others. So be able to identify those patients who are not getting benefit and may have benefit with a cellular therapy, that's the main point of the work that we are doing right now, and try to leverage on the existing data, on the amount of data that every one of us is collecting through the studies.
Yes, yeah.
Yeah, no, I would agree with Alessandro. I think that now the aim is, especially if these assets are moving in earlier therapies, where, as Alessandro correctly said, there are standard therapy, maybe the focus is to, let's say, to select the high-risk patients where there are some genetic and molecular, I would say, signatures that are not responding to the standard therapy, and this could be in one space when so innovative therapies can be applied, I would say. And on the other side, I think that now in Europe, with the big registries, and I think France is representing a very good example how the registries can deliver a lot of information. With the national registries you have here, but Europe is following, is also helping, let's say, to identify with big data, maybe cluster of patients that can be, let's say, very good responders versus others. So I think that the match between, and the technology can help, between big data analysis and molecular signatures could be also a field, and the future field, how to work all together. I think private, so sponsors, industries, and academic centers.
Okay, so there are some very modern approaches that you talked about this morning. The idea of add magnetic nanoparticles to follow CAR-Ts into the distribution with RMI. Is it something that could be imagined in the future?
For sure, of course, we need to validate that using preclinical models. So, I mean, the idea, and we have an ongoing collaboration with Laurence Gazot also, is to use magnetic nanoparticles and to load the CAR-T cells with these magnetic nanoparticles, inject them into an animal bearing a tumor, and then using MRI to follow the biodistribution. I mean, it's very important where the CAR-T cells go.
That's right.
We know that in patients, the first organ where the T cells are going it's not the tumors, it's the lung. They are sequestered into the lung for 2 days. So it could also raise very important issues about toxicities in the lung. And the other aspect, of course, since these nanoparticles are magnetic, then you could add magnetic fields or a magnet in order to retain or attract
to the tumors.
Because we know, and we heard this morning about the different obstacles preventing, at least in solid tumors, really preventing the CAR-T cells to home correctly, to infiltrate correctly. This is really a major obstacle. So having some strategies able to attract T cells would make a lot of sense.
Why not injecting directly the tumor?
It has been done, of course. I mean, for example, for brain tumors, Even, I mean, I could refer to a publication done by a colleague at Stanford, or Imaginer, Christian Michael, treating some children with glioma, some kind of very aggressive glioma, and they infuse the CAR T cells in the brain, and they actually evidence very promising result, although not enough to cure these poor children, but really very encouraging results. So,
do you work on that kind of strategy to inject directly the tumors?
Well, as of now, we are not focusing on that because that's not an approach that is feasible in many diseases, where you have one lesion that you can easily target and inject your cells. So the beauty of the cell therapy is really to leverage on the natural tendency of your immune system to fight against cells that can be distributed everywhere in your organism. So this is the main point of cell therapy, that you give just an injection and then the cells knows where to go and then they are activated.
When it works.
But of course, the main, I agree with, especially with solid tumors, the infiltration of the tumor is one of the main difficulties and barriers that we are facing. So how we can prevent that and overcome that issue, and how we can engineer these cells to overcome that issue.
Okay.
Yeah,
Yes.
yeah, I agree, and Johnson & Johnson has recently been embarking on something called interventional oncology, meaning that we would like to treat the tumor at the site of the tumor. And of course, for cell therapy, I mean, it's still a long way, but we're envisioning how to, instead of using the systemic delivery route of drugs, how we can inject and deliver the drugs really on-site using robotic platforms that, of course, we use from our other sector, Johnson & Johnson MedTech. So we're really joining forces now with the MedTech sector in order to come up with innovative solutions to deliver intratumoral.
I think artificial intelligence must be helpful to help design.
Absolutely, and that is throughout the whole portfolio, of course, that AI is making its introduction in not only in drug discovery, but also in various parts of data sciences and analysis of big data and real world data. So absolutely.
Great, thank you. Do you have questions for panelists? I see Mark is ready, but if you have, okay, you want to start? Yeah, take your mic, thank you. Yes, normally. Can you hear? No, okay. I think the mic, try again. I think, is it connected? Our mic's connected on the
It
desks?
works, no?
Yeah.
Okay.
Thank you.
Many thanks for the very interesting discussion. I'd like to ask a question about dose reduction and potential for cost reduction related to that. We've seen a number of studies, including the T-charge protocol data that was presented earlier today. Also, the studies from UPenn where decreasing dosage have been shown as effective, if not in certain cases more effective, given reduced time of expansion and arguably less exhaustion of the cells. So I'd like to ask the question this way to the panelists. Do you see any lower limit to the dosage that could be achieved and still be effective? And in that way, to reduce the cost of the treatment?
Very good question. Who wants to answer?
Maybe
Yes,
again,
yeah.
using the right type of cell with the right capacity to expand could be one solution. If you can use the right type of T cell and you get the right percentage of TSCM, meaning cells who are really fit, numerous, able to expand, we may participate to both improve efficacy and diminish cost and doses. The other solution, I'm sure, it's about scaling and manufacturing, and I'm sure there is a lot of hypotheses and scenario and methodologies investigated on the stage.
Yeah, I think it's part, I would say, of the solution. It's not really let's say, the solution in terms of lowering the cost, because I think it's also costs are related to other variables other than, the dosage and the administration of sometimes you need to have, to give the possibility to the patients and, to be treated. And then on the manufacturing side, that you need to be sure that you are developing and having the right cells to be administered. So the cost is also related to some, I would say, possibility that you have a second bag, for example, or others able to be administered to the patient. Is part of the solution, is not totally related to the cost. My point of view is that with therapies that are one shot, theoretically, always you can avoid other type of treatment. So the cost containment is when you have the possibility to manage very well in a health system where companies and institutions are aligned. Maybe there are other collateral costs that you can maintain. It's not only the cost of the product itself. And maybe the intervention and the alignment between companies and public is also to see how to harmonize those treatments on the field, I would say. So it's a complex questions where there is not one right, I would say, answer, at least my personal opinion.
Okay, yeah. Alessandro?
I think the reduction of the timing of the cell expansion, it may not be the solution for each of the products, because for the available products, we know that the dose count because it's important, and there's a dose-dependent response. So may not be applicable for all the cell therapy that are developed. For some of them, yes. And of course, there's a lot of work to focus on how we can improve the in vivo expansion. And that's what they are leveraging some of these platforms. That, of course, it's important. Overall, on the cost, I don't think that the main driver of increasing the complexity and the cost of the production is like 2 or 3 additional days in incubation to just grow more cells. It's all the complexity around the production system. But of course, it's definitely something that, as you can hear, someone is already looking at, and we are also looking at how we can reduce, because, for example, even in our product that we are developing in autoimmune disease, we are taking a new platform that is reducing the time of production. So those are all points that we are taking, but may not be the response for everything.
Thank you. Other questions? Yes.
The question for Karin. Amazing technology around thymus. I was wondering, you're still talking about adult thymus, like when you're infusing them, they're going to whatever thymus you have left over, and whether cytokines such as interleukin-7 could be useful, because there are thymic implantation that has already been FDA approved, but they failed to develop naive T cells. So I was wondering, like, what are your thoughts around that?
It's a very good question. Thank you. So we have demonstrated both in vivo, but we have also some first data in men showing that there is a crosstalk, actually, between the pro-T cell and the thymic epithelium. And you're right, IL-7 is very involved there. But it seems like our cells can increase thymic epithelium activity, and especially medulla, which is in charge of educating and differentiating fully the pro-T cell into effective T cell. So the first data we have seems to show that we are able to provide mesenchymic immigrant even in elderly patients, meaning that there's still sometimes tissue, and there could be a way to wake up thymic activity. That's what we aim to demonstrate. And that was actually the reason why the Bill and Melinda Gates Investment Strategic Fund did invest in our company. They are not, they did support an oncology phase 1-2 in adult leukemia, but they are not really interested in oncology. They are really intrigued on whether or not our technology can reverse thymic involution.
And you talk about investment. Bill and Melinda Gates Foundation investing in your company, that's right?
Yeah.
So that's noticeable. Thank you. Okay, other questions? Yes, please. Can you turn on the mic, please?
Oh, yeah, sorry. I have a question from Emmanuel. Actually, you talk about the magnetic tracking of the CAR T cells and using the mouse model to do that. So my question is, like, is there any way that you use the data from the mouse model that you use for toxicity to find the relevance of the outcome, that how it could be in the human? Because you told me that the model is not like efficient enough to compare the toxicity in humans. So how you actually, what would be the readout relevance to human when you use this tracking of the CAR T cells to understand the infiltration of them in the human?
Well, I think these are two different aspects, the tracking, the biodistribution. And again, also, if we're able to attract T cells to the tumors, we may actually prevent the T cells to home too much time into the lung or other healthy organs. So it would make sense also to developing such approaches. And concerning the other models to test for toxicities, then I will go directly to human ex vivo 3D models, especially for the on target of tumor toxicities, having a tissue that is susceptible to express the target and test for CAR T activation and potential cytotoxicity, as we are doing actually, testing a large number of different CAR T for the T2Evolve consortium.
Okay, thank you. Other questions? Mark?
I would like to go back to the approach of CAR T in an allogeneic setting. So clearly this is a very promising approach, many challenges naturally. So first one, avoid alloreactivity of CAR T, so you have to knock down the endogenous receptor. Avoid rejection of allo CAR T, and then what could be the most promising approach? I remember several early cellular approach in infectious disease, where people used to select cells expressing commonly expressed HLAs. You have the possibility to knock down HLAs. It's completely doable now, but basically then you will trigger rejection by NK cells. So what would be your most promising strategy to optimally use CAR T?
Great.
Okay, thank you very much. It's a fascinating question and not a $1 billion question, as you would say. We do believe that, of course, what has been done today in allogeneic CAR T is amazing, and the addition of the cells and of the HLA or TCR does allow to get those cells into the body of the patient and avoid GVHD. We do think that the Thymus can do this job maybe more safely first and as efficiently. At the maximum, we would like to avoid to edit those cells. So we are developing with our R&D team, and they are very talented, Dr. Olivier Negret is around, an algorithm to choose among healthy donors, homozygote donors, and the more relevant HLA able to be used in an off-the-shelf biobank, so that 20 biobanks could cover somewhere around 60% of one population. That would be, to us, our goal, meaning it's a biomaking approach.
It's your purpose? That's what you'd like to develop yourself?
That's what we are developing.
You're
yes.
developing right now, okay. Good.
I think this is the main challenge for allogeneic CAR T, because we want to have a product that can survive and be active like autologous CAR T. I don't think that what we have right now in the allogeneic field is the response. There's much more work that needs to be done. And I think you touch on an important point on how we can avoid the rejection. So there are, and these require multiple engineering. So the aspect of, and the difficulties of entering into the allogeneic field is really how many additional engineering you need to modify your cells to avoid GVHD, to make these cells persistent and get the right target. And so there are options already, also, for example, expressing HLA-E to avoid the NK rejection and so on, but of course increase the complexity of the engineering and how we can simplify that part of the engineering and make it sure that we are producing a safe product, and how we face all the regulatory hurdles that we have with multiple engineering of a single cell.
And one point, if I can add, is how much we need to engineer those cells in the reality would reduce the cost at the end. Because we start that we say, oh, maybe potentially this can be a cost reduction.
Yeah.
We are seeing that on the paper, yes, is an ideal aspirational, but at the end, if we need to go deeply in engineering a lot those cells,
It will cost a lot.
at the end, the cost, I don't know how much will be the difference at the end. We need to evaluate.
So it means that you don't look at the strategy? You don't
No, this is what we are looking also at the end, but at the end, we need to see what currently we have. We are working, with also academic partners in terms to see what could be the best solutions. But
you let them do the hard job and then you see afterwards?
Together, I would say. Together, this is part of my job, is to make those kind of deal and partnership. But again, I think that as a scientists and clinicians and companies, we need to understand that aspirationally, yes, this is a very good science to be, and there are solutions, and there are very good solutions, but how much we can intervene, and then the cost is not the reality, the lower we can have, is also something to evaluate.
And the exhaustion of the cell, because they don't like to be edited. So again, I'm not saying that progenitors are the solution to everything. It's another, way to try to tackle the problem, and we will need to collaborate to find the right way to make it efficient in the body. But what is true is that there is cost for all those engineering steps, but there is also the fact that a T cell needs an HLA and a TCR to live 7 to 10 years in a human body. If they don't have it, they don't know how to interfere with their environment. So we need to find the right way to tackle cost, exhaustion, safety, and work all together, each of us bringing one piece to the jigsaw to make it happen for the patient.
Yeah, I think the key is collaboration in this. Absolutely. And
You'll agree on that, I think. You'll agree on that point. Yeah,
yeah, absolutely. And actually, it's my job as external innovation, to look for partners that are agile and nimble to tackle these sub-questions of the big questions, of course. Mm-hmm. And indeed, we are also putting a lot of effort in this allogeneic approach. And again, the engineering is very complex because you want them to be immune stealth, but you still want them, of course, to be immune reactive at the same time. So there's a lot of engineering costs that comes with it. And again, and I bring it up again, we should also look at how do we want to make these T cells. When we do talk about T cells, that is a variety of cells that still can be, let's say, we can pinpoint towards alpha beta or gamma delta, which might have already features that we don't have to engineer up front. So.
Yes, typically, for gamma delta T cells, naturally. But, you have two ways to address this. The first one is typically gamma delta cells, like NK cells, might have NK receptors that would make them more prone to detect stress-induced ligands and so. But also, and I think that this has been addressed by some companies, but not that much, the possibility to transduce gamma delta T cell receptors that are directed against conserved antigen that are upregulated on a wide variety of solid tumors. And I think that this is really an interesting opportunity because these receptors seemingly show limited toxicity. They show strong no alloreactivity, strong reactivity against a broad array of tumor cells. Mm-hmm, mm-hmm. And I think that this is really a track that could be...
Absolutely, fully agree.
Yeah, and we are looking, for example, at that. But as we were saying, I think there are so many things to look at the same time, and that's why partnership are extremely important. So that's what we are doing at BMS, that we have we are trying to create and be at the center of an ecosystem of innovation and try to create strategic partnership with different actors from the academia to biotech that have the most recent cutting-edge technology that can give us multiple strategy to address all these questions that are a lot of questions that need to be addressed at the same time.
So, B2B sessions are here for that. So in a few minutes now, from now, during 2 hours, you'll get occasion to discuss of these points. Thank you very much to all of you
Thank you.
for your participation. Thank you.